cell, it's called the fundamental unit of life. Why it's called the fundamental unit? Because it's the smallest structural unit to build up a multicellular organism, and also it's the smallest functional unit to carry out all biochemical processes essential for life. Now, if we pick up the cell organelles and put them outside the cellular organization, then they cannot survive or perform the biochemical processes. It clearly proves that the cell is the smallest functional unit for life. Now, the type of cell. Basically, the cell is divided into two categories based on the type of nucleus present within the cell. The nucleus, or called karyon, is a cell organelles which is called the brain of cell because it controls the metabolism of that cell. Now, as the karyon, that is the nucleus, is primitive and ill-developed, that is poorly developed, such type of cell having this primitive nucleus, as for example bacterial cell, termed as prokaryotic cell, and those cells with well-developed nucleus, as for example all the animal and plant cells, contains developed nucleus. So, cells having developed nucleus or modern nucleus is called eukaryotic cell. In the next page, that is page number 2, there is a brief but clear idea according to the presence or absence of the cell organelles among these three types of cells, that is prokaryotic cell and eukaryotic plant cells and animal cells. Now start with the outermost covering layer of cell called cell wall. It is present both in prokaryotic and plant cell, but the animal cell don't have such type of cell wall. Point 2. The cell membrane. It's a living lamella and allows the cell to exchange the materials, and other functions of cell membrane and cell organelles will be discussed later. Now, the cell membrane must be present in all living cells. Without cell membrane, cellular organization cannot be developed or cannot be present, because the point number 3 depicts the cytoplasm, that is the plasm or fluid present in the cyto or cell is bounded by that particular membrane called cell membrane. So if cell don't have any membrane, it will lose its plasma, and as it loses its plasma, then the cellular organization will be disrupted. Cytoplasm contains all the organelles in it, so it is present in three types of cell. Now point 4, well-developed nucleus. It is not present in prokaryotic cell, and this ill-developed nucleus is called nucleoid. But the developed nucleus is present both in plant cell and animal cell. Point 5. The ribosome, which synthesize protein in cell and termed as protein factory, is present in prokaryotic cell, eukaryotic cell. But their nature or types are different. In prokaryote, they are 70S type, and in eukaryotic plant and animal cell, they are 80S type. S is the Wedberg's unit. Point 6. The membrane-bound cell organelles. Prokaryotic cell do not have any membrane-bound cell organelles, but eukaryotic cell contains such type of cell organelles as A, B, C, chloroplastid, mitochondria, and endoplasmic reticulum. All these three are membrane-bound cell organelles, and they are represented here according to their presence and absence in plant and animal cell. Now, Golgi body. In plant cell, it's termed as dictyosome, and in animal cell and plant cell, the Golgi body present, but in plant cell, they are not termed as Golgi body. Rather, they are termed as dictyosome. But in animal cell, they are termed as Golgi body. Now, the centrosome, and cell organelles that lacks cell membrane. It is absent in prokaryotic cell and plant cell, but present in animal cell, help during the cell division. And type of cell division in prokaryotic cell is amitotic, but in plant cell and animal cell, the division is mitotic and meiotic, that is equal division and reductional division. Now come to the next page, page number 3, where the features of prokaryotic cells are written on the right hand, and a figure of prokaryotic, that is bacterium cell, is attached at the left end of the page. The features clearly shows that the cell is very simple, having a cell wall composed of peptidoglycans. Peptido means protein and glycans means sugar. So the cell wall is composed of a complex structure of protein and sugar. Beneath the peptidoglycan cell wall layer, there is a living plasma membrane. The plasma membrane is the same structure present in all the cells, whether it is eukaryotic or prokaryotic. The cytoplasm lacks any membrane-bound cell organelles and well-developed nucleus absent. And the structure where double-stranded circular DNA is packed in the cytoplasm is called nucleoid. It's a dense area of cytoplasm. Ribosomes are 70S type. There are some extra chromosomal, that is devoid, or they are present outside the nucleoid, double-stranded circular DNA in the cytoplasm. So outside the chromosomal structure or nucleoid, the double-stranded circular DNA, which carries some special type of genes, is called plasmid. The nucleoid is a structure that lacks histone protein, and hence, in prokaryotic, chromosomes are not formed. Now, the mesosome. Mesosome is the infolding of plasma membrane. It helps in replication. Replication is a process to copy its DNA, or replication is a process to duplicate its DNA. Mesosome also involved with cell division and cellular respiration. Now, the outer layer or slime layer, also called the jelly layer, that prevent the bacterium from desiccation, that is dehydration, and also help to attach with substratum, may present in some cases. The locomotory organs are present as the flagellum. The fimbri or cilia performs for adherence, and the pili, the hollow protein-built tubes that helps in conjugation. So it is very simple structure and smaller in size in comparison to eukaryotic cell. Now come to the next slide, that is fourth slide, the virus. Features of virus. Viruses are acellular, that is devoid of any cellular organization. They contain as the DNA and genetic material, DNA or RNA as genetic materials, and this genetic materials is covered by protein coat called capsid. Here a phage virus, the figure of phage virus is discussed or pointed, and it shows a head, tail and a base plate. The head contains the hexagonal capsid, that is the covering, and this hexagonal capsid is composed of small triangular facets, that is the piece of capsid called capsomers. Actually, in 3D model, it's not hexagonal, rather it's called icosahedral. Within this capsid, the double-stranded DNA is present. The head is attached to the long tubular tail through collar, and the collar contains fibrous whiskers. Now the tail is wrapped through contractile sheath that may contract during infection to bacterium. The tail is attached to hexagonal base plate. Each corner of the base plate contains tail fibers, and beneath the hexagonal plate there are spikes at each corner. This virus is called bacteriophage or phage virus because they reproduces or they infect the bacterium. When they attach the bacterium with their tail fiber, they pushes the DNA through its spikes and tail in between the space of bacterial cell wall and cell membrane. Now the DNA will take charge of metabolism in that bacterium, and later they will produce same phage particles within that infected bacteria, and after its completion of life cycle within the host cell, that is bacterium cell, they rupture the bacterial cell and huge number of phage copies are released. features of virus clearly proves that they are living only inside the host cell and behave dead outside the host cell. They are crystallizable, shows no growth, development, and any biosynthetic pathway. Only they are composed of nucleocapsids. And virus is very tiny and only can be seen through electron microscope. In the fifth slide, that is page number 5, the features and the figure of eukaryotic cells are represented. The feature shows that well-developed double membrane bound nucleus present within the cell. Genome contains both the histone and non-histone proteins that helps to form the chromatin, that is the thread present within the nucleus, and chromosome, that is rod-shaped structure present during cell division. And the ribosomes are 80S type, but as they contains membrane-bound cell organelles like the mitochondria involved with cellular respiration, that is oxidation of food to produce energy, chloroplastid performs in photosynthesis in plant cell. They are membrane bound and they have their own DNA as well as they have their own ribosome to synthesize their own protein, and the mitochondrial ribosomes and the ribosome in chloroplastid, they are not 80S type, rather they are 55S type. In this page, the figure of animal cell and plant cell clearly represent the shape, size, number of cell vacuole, the presence and absence of different cell organelles in between these two eukaryotic cells, the presence of centrosome, and other differences. So, you can understand the difference in between plant and animal cell. They are pointed images for your exam purpose or your understanding purposes, and the different cell organelles, their occurrence in cell. So go through this chapter, study further. This is the schematic or mapped model prior to your study through your textbooks or anything or any study material provided to you by any authorities. You go through this mapped discussion and then follow your own study material. Thank you. Cell. It's called the fundamental unit.